Hello everybody and welcome back to this short course of Bioinorganic chemistry. We have been discussing about our life with dioxygen. As one can see that the composition of earth crust where this dioxygen is the most abundant and composition of air where is dioxygen concentration is around 20.9%, the huge dioxygen concentration, and most of the oxygen consumed by aerobic organisms is used to produce energy in a process referred to as oxidative phosphorylation, a series of reactions in which electron transport is coupled to the synthesis of ATP and in which the driving force for the reaction is provided by the 4 electron oxidizing power of dioxygen. As you can see that O2 converting to water with the help of 4 proton and 4 electron with a E0 value of 0.815 volt is a measure of great oxidizing power of the dioxygen. Once you look at the redox chemistry of molecular oxygen, if the 4 proton and 4 electron is not added all at a time, then you can get the stepwise reduction which eventually leads to O2 minus, O2 2 minus and OH radical, which of course converts to water eventually. So, we have discussed all these in details in my last lecture. However, if you look at reduction potential of all these steps, then what you can find that first reduction of dioxygen to produce O2 minus is a very unfavorable reaction. E0 is minus 0.33 volt and this 1 electron reduction of dioxygen to O2 minus presents a barrier that protects vulnerable species from the full oxidizing power of dioxygen. So, this is the thermodynamic barrier. As I discussed in my last lecture, dioxygen is in triplet state, while the organic molecule this is in singlet state. So, it will not react and this is because of spin forbidden. So, triplet state or dioxygen has to convert to singlet state to react with an organic molecule. And if you look at this diagram, as you can see that this is the ground state of dioxygen which is shown over here as a triplet state, and this goes to singlet state which is in first excited state which is in delta state with the help of 22.5 kcal per mole, and then the second excited state it is in sigma state which require 37.5 kcal per mole. So, once you provide energy, then and then only dioxygen can convert from triplet to singlet and thereby it can react any organic molecule. So, this is a kinetic barrier which dioxygen need to cross for its reactivity. Now, we will talk about the survival of life, all about bioinorganic chemistry. Actually, two most important bioinorganic reactions, or rather in other words, two most important reactions in biology is one is, as you know, photosynthesis, another one is respiration. In photosynthesis, what is happening? In presence of light, this carbon dioxide and water forming carbohydrate. This carbohydrate during the respiration in presence of oxygen convert to that huge amount of energy which we use in our day to day activity. For example, how this food is converted into energy. This glucose C6H12O6 plus 6O2 eventually converted to 6 carbon dioxide plus 6 water plus release huge amount of energy in the form of ATP. ATP is adenosine triphosphate. As you can see that this is adenine, this is ribose sugar, and there are 3 phosphate group. This is 1 phosphate, 2nd phosphate and 3rd phosphate, that we call it triphosphate. Now, this phosphate 2 phosphate is actually linked with POP bond, and these bonds when it breaks releases energy. Actually, when you need energy, ATP convert to ADP, adenosine diphosphate. You see that 2 phosphate, phosphate 1, phosphate 2 and another phosphate PI, inorganic phosphate, and releases energy which we use for various activity all the time. And then again when we generate energy, then it will be stored by converting ADP to ATP, adenosine diphosphate to triphosphate. When we have more energy to store, what is happening? That POP bond will be formed, just like ATM. If you have a ATM card and you can store your excess money you have, or you can take out some of this money when you need it. So, ATP is universal currency for energy in living system, and when you need it, ATP converts to ADP plus inorganic phosphate PI and releases energy, and delta G0 is minus 7.3 kcal per mole, which is lot of energy, and this energy is utilized for our day to day activity. Say what I am talking in front of you, also I am spending energy. Most redox reactions involving organic molecules occur in the range of 0 to minus 400 millivolt. The oxidation of the biological fuel like carbohydrate involves reactions in which electrons are passed through the electron transport chain until eventually hydrogen and electron enters the NAD plus NADH couple. You can see that this is what is chain reactions. So, electrons comes to ferredoxin, and this is at a potential minus 0.4 volt, and ferredoxin gives the electron to NAD, and once it gives to electron to NAD, ferredoxin get oxidized and can take another electron from the food source. Now, NAD when it gets one electron, it becomes reduced, and once it becomes reduced, spontaneously gives an electron to the flavoprotein, and flavoprotein get reduced, NAD get oxidized. Flavoprotein gives electron to cytochrome B, then cytochrome B will be reduced, flavoprotein get oxidized, cytochrome B gives electron to cytochrome C, then cytochrome C get reduced and cytochrome B get oxidized. Cytochrome C then gives electron to cytochrome AA3, and where all these electrons comes to O2. You know that O2 plus 4 proton plus 4 electron gives rise to water. It requires a potential of plus 0.815 volt. So, this potential is required, and biology gets this potential, and also all these electrons like this spontaneously without any problem. You need 4 electrons all at a time, and of course, protons would be there, and this dioxin converted to water. So, this is what is happening, that dioxygen at this potential of 0.815 with the help of 4 proton and 4 electron converts to water, releases energy, and this is what is happening in cytochrome C oxidase. And as you can see, 2 iron 2 heme center and 2 copper center convert to 2 iron 3 heme and 2 copper 2 plus, and the dioxygen 4 proton 4 electron converts to water, and this releases lots of energy. This is the terminal step of this oxidation process. If we focus on this heme and copper unit, so what is there? There are 2 types of copper, as I have said. Copper A, which is a dimeric copper, as you can see, 2 copper unit are here. This is 1 copper, this is 1 copper, and cysteine sulfur bridges between these 2 copper. Now, copper B, as you can see, the 3 histidine are ligated to this metal, and also 2 types of heme, one is called heme A and heme A3. 2 histidine moieties are there in heme A, which is 6 coordinated, and here in heme 3 it is 5 coordinated. So, heme A actually responsible for electron transfer, because it is already coordination number 6. Dioxin cannot bind over here, but heme A3 it is 5 coordinate iron center, where dioxin or other species can comes and binds in. So, let us look very carefully that how this enzyme works. Cytochrome C gives electron to copper A and cytochrome A, they get reduced and they transfer this electron to cytochrome A3 and copper B, and then this copper A and cytochrome A get oxidized, and this is the place cytochrome A3 and copper B where actually dioxin binds and converted to water molecule in presence of H plus ion. If we look at that cytochrome A, it is already 6 coordinated. So, there is no place for dioxygen to coordinate, whereas here you see dioxin can coordinate to iron center and dioxin can coordinate to copper center as well. So, this is what is the flow chart, like 4 electron. So, this comes to first copper A and copper A getting reduced, then copper A transfer electron to heme A and heme A get reduced, iron 3 becomes iron 2 and this copper 1 gets oxidized to copper 2, which takes another electron. So, all this electron passes heme A to this heme A3 and copper B, this region where actually dioxin binds, where dioxin needs 4 proton 4 electron to reduce it completely to water. Indeed, this is what is happening. This is what is the dioxin reduction center, and this is what is the chemical reaction 8 H plus in plus 4 electron plus oxygen gives rise to 2 H2O plus 4 proton out. And as you can see, the structure of iron copper center is shown over here. It is a heme, porphyrin ring is there, and this is histidine, and it is a 5 coordinated species, whereas similarly copper it is 3 coordination, 3 histidine is ligated to copper B. So, there are empty back sites where dioxin indeed can comes and binds. Now, we will see that how it binds dioxin. So, this is the structure of cytochrome C oxidase where it binds dioxin. You can see that this iron and copper binds a dioxygen, and there is a huge protein chains which is wrapping this small iron copper unit, and if I remove this protein chains, you see now that this iron copper unit binds dioxygen like this, and this dioxin is O2 2 minus. Resonance Raman stretch confirms that this binds in O2 2 minus. So, this is the crystal structure of He-3 copper B sites of the fully oxidized cytochrome C oxidase. Now, question is that how dioxin can binds, whether dioxin binds first to copper, then what would happen, that it can forms like this O bond, or dioxin first can bind to iron center as well, and it forms this iron 3 O2 minus, as we already studied. So, however, both these forms gives rise to this copper iron O2 2 minus, and this is the product which is forming. It does not matter whether it first binds to copper or first binds to iron. This question is still debatable. Most probably dioxin first binds to iron center and convert to this species, and as I have said that this superoxides, this O bond dissociation energy is lowest, where this O bond easily breaks and converts to water. We will see the mechanism right now. So, the overall chemical reactions is O2 plus 4 H plus 4 electron gives rise to 2 H2O. Now, this is what is iron copper unit in the resting state, where it is iron 2 and copper 1, and dioxin probably will bind to iron center, as I have said in previous slide that it also can bind to copper center as well, but possibly iron center is preferable to bind dioxin, and you know that iron 2 will be converted to iron 3 and O2 will be converted to O2 minus, and once it forms O2 minus, then O2 minus this also binds to copper, and then copper 1 will convert it, will oxidize to copper 2. So, iron copper peroxide complex is being formed, and with the help of 2 proton and 1 electron, this forms iron 4 oxo and copper OH2, and then after 1 electron reduction, this iron 4 oxo converted to iron 3 OH, and this then react with 2 H plus, which would convert to 2 molecule of water and gives rise to iron 3 copper 2 fully oxidized species, which will reduce by 2 electron, gives rise to this iron 2 copper 1, and which is now again ready to bind dioxygen and start the catalytic process once again. So, overall reaction, as you can see that here 1 electron, 2 electron and 2 electron. So, 4 electron and 4 proton, 2 H plus, 4 proton gives rise to 2 H2O and releases huge amount of energy, and which we store it. The mechanism is so foolproof that you will not get any partially reduced species. However, nature also can do mistake. So, instead of getting water, there is a chance that you can generate the partially reduced species, like it can form O2 minus after 1 electron reduction, which is, as you know, that it is called superoxides. After another electron reduction, it also forms peroxides O2 2 minus, it also can form OH radical. So, all these species are highly poisonous for our body and in biological system. They are called biological poisons, and these are collectively known as reactive oxygen species, ROS, reactive oxygen species, O2 minus, O2 2 minus, OH radical, all these are actually responsible for various problems related to aging. Now, let us little bit talk about on that topic. Now, Hermann first proposed this free radical theory of aging as early as 1956. Now, aging and age related diseases result from ROS mediated oxidative damage of lipids, protein and nuclear and mitochondrial DNA molecules. Okay. These are all results of the radicals. You know, radicals are very dangerous in biology. Radical reactions are indeed very difficult to control. You can see the effect of reactin oxygen species and aging in these two pictures, and you see that this sides, as you can see that this is clear effect of free radical and aging problem, and you can see this part and also very clearly. So, without aging problem, another one is once you have this reactive oxygen species and how it creates the problem on your face, on your neck, on your eye, on your body. There are many examples, just 2 examples I have shown you to attract your attention over here. Now, let me talk a little bit about antioxidant. Now, antioxidants are molecules capable of reducing the cause or effects of oxidative stress. Oxidative stress can be caused by environmental factors, diseases, infection, inflammation, aging, as I have just shown that ROS production, reactive oxygen species production. ROS or reactive oxygen species includes free radicals and other oxygenated molecule resulting from these factors. The body produces some endogenous antioxidants, but dietary antioxidants may provide additional line of defense. Flavonoids and other polyphenolides, vitamins C and E and carotenoids are the most common dietary antioxidants. And these days there are many herbs and botanicals also contain antioxidants. Now, these are the foods where you have antioxidants. Okay. Huge amount of antioxidants like green tea, onion and these oranges, tomatoes and all sorts of things. There are full of antioxidants, and we can use those antioxidants to get rid of age related problems also. As you can see, one can protect the skin from the effects of such reactive oxygen species just by applying these antioxidants time to time, and thus one can stay teen even at very old age. This has been very popular therapeutic techniques at home as displayed in this slide. In summary, I have discussed today how dioxin is reduced to water by the use of 4 protons and 4 electrons in cytochrome C oxidase, which is the terminal member of the respiratory chains. The reaction eventually responsible to produce huge amount of energy that we use every day. However, incomplete reduction of dioxygen produce reactive oxygen species such as superoxide, peroxide and OH radicals, which are indeed extremely poisonous for our body. Aging and age-related diseases also result from reactive oxygen species mediated oxidative damage of lipid, proteins and nuclear and mitochondrial DNA molecules. I will discuss in my next lecture how our life is fully protected from such reactive oxygen species. Thank you.